Good morning. I want to thank you for joining us this morning for our practical approach to antibody labeling. This webinar is from Molecular Probes in Eugene, Oregon. We're part of a life technologies company. I'm the presenter today. My name is Judy Berlier. I'm a staff scientist here. And I wanted to let you know that also on the line, available to answer your questions as we go, is one of our technical support scientists, Olivia Holdova, and she will be able to answer questions during the webinar if you happen to have any. So I guess we'll go ahead and get started. The topics that we're going to cover today are going to be common labeling techniques, labeling kit options, application-specific hints, DIY answers, and custom conjugates. So it's kind of a generalized flow to get an idea of what we're doing, what we're talking about, and then how you want to go about doing your antibody labeling. So the next slide, getting started. Normally there are things that a person that wants to use one of our labeling kits needs to ask. They need to know, is their protein purified? And I'm saying protein because a lot of people, some of our kits are actually designed to label antibodies as well as other proteins. So in this case, you need to know if your protein or antibody, either way, is purified. And what I mean by that is it a purified protein, or is it something like an ascites, or something that has a stabilizer? Is it in PBS? Is it in tris, imidazole, glycine? Does it have any primary amines? Does it have a high amount of biocides or any other stabilizers, protein-based like gelatin? These are the kinds of questions that somebody needs to ask themselves because it's going to make a difference in how you're going to go about your labeling. The next question that you're going to ask yourself is how much you want to label. So we have kit sizes that are available for different amounts of protein. And so if you had a milligram of protein, you would want to select one of our kits that's designed to label a milligram, as opposed to if you had 10 micrograms, you would make a different choice. And then also, do you want to label your antibody and use it, or do you need to optimize your antibody and your labeling conditions? And so those are questions that normally people ask themselves when they're making those decisions. So we'll talk about antibodies, even though that's not the only thing that you can use for the kits. But basically, for an antibody, there's two portions. You have the two FAB regions, so it's an FAB2, or a whole IgG, or a fragment, depending upon if you cleave it. You can make, with papain, you can make an FAB fragment. It's a monovalent fragment that has one antibody binding. If you use pepsin, you have a bivalent FAB fragment, so you have two binding sites, which gives you better affinity of labeling to your target. And then you also have the FC region, where you can actually have, FC receptor-mediated activity. So it's not necessarily a specific binding, but that's important in an antibody. So, and you see also with the antibody, you have disulfide bridges, where people actually will use those to cleave and use a free thiol. So there's several reasons why we want to look at it. If you're doing amine labeling, you can have a lysine, which is the specific amine that you're usually labeling when you're doing a amine labeling reactivity, and it can be anywhere on the antibody. Usually on the FC portion, you have glycosylated areas, and so a lot of people want to use that part for targeting. That's possible. It's something that's becoming more possible, but what we're really going to talk about mostly today is more common ways of labeling. So moving on to the next slide. So how do we do our labeling? So we usually want to know if we're going to be labeling amines, the thiols, which is if you're cleaving your disulfide bonds in your antibody, or you're putting a thiol on your protein, you can use that as a labeling handle. You have to have some reactive group that you can modify. There are other groups. You can use aldehydes, ketones, alcohols, sugars, and you can also use click chemistry. So our primary mode of chemistry that people use is usually amine labeling. And the reason why is because it's easy, it's robust, it happens at pretty common conditions, room temperature labeling, it's fast, it's efficient. The conjugates that you make are very stable. The things that you have to know is that in order to have your target amine be able to react with your dye, you have to have it deprotonated. So you usually raise your pH with sodium bicarbonate, and then you add your excess dye. You do a purification versus a size exclusion resin, and you purify your product, and it's labeled. This isn't necessarily something that you have to do, but our kits basically have the whole package. So if you need to know what you're labeling, and you need to know what you're getting so that you can use an optimization protocol, you need to know what kind of labeling you get. If you don't need to know, we are going to talk about that you can use, in many cases, the kits and not even use the column. If you need 100% recovery, you just want to use it. If you're going to do washes in your applications, it's perfectly fine not to use the columns. So in this slide, we're basically showing the common amine reactive dyes. So esters are very efficient modifying reagents. We have them in many different types of fluorophores. So they're all ester reactions where they're reacting with the deprotonated amines on your protein, and they form a stable amide bond. And here are some other ones. They have different leaving groups. That's really the only difference between any of the amines or the esters that we're looking at, is that their leaving groups are different, so they have different efficiencies of kinetics, but basically they all form the same amide bonds that are very stable. And then we also have isothiocyanate dyes like TITC and TRITC, which are pretty common. They're a little bit less stable dyes, or less stable conjugates, so the thioureas are still stable. They're covalent bonds, but they're not quite as stable as an amide bond. And then we also have sulfonyl chloride reactive dyes, which also react with deprotonated amines on your protein. These form a sulfonamide. They're also stable. We don't have that many choices available. Texas red sulfonyl chloride is a pretty common one that people use. And then the other ways that we go about making conjugates are things like thiols. So the reason why people use thiols is usually because they're rare. So usually you see disulfide bonds, or somebody can add a specific thiol when they're doing a mutation, so they can have a specific site that they're always going to be able to label, and they're never going to over-label. And so in that case, and we do have proteins where people actually have made site-specific mutations so that they can have specific binding. And other ways that you can do it is to cleave your disulfide bonds in your protein. And a lot of the common ways that people do that is by using TSEP, beta-mercaptoethanol, or DTT. These just cleave the disulfide bonds and leave free thiols. In the same case that you do with the esters, you use an excess of the dye, and then you purify away your conjugate, getting rid of the free dye. So thiol reactions very similar in terms of the reaction, except for that you're using the thiols on your protein, and you react with a maleimide or an iodocidamide, and you get a stable thioether bond. And this is common for labeling with, any thiol-containing protein. If you reduce it and want to label it with these two, they're stable bonds. They are also very common for using when you want to make heteroconjugates. So if you're doing a protein-protein conjugation. So this could be something where you're using a R-phycoerythrin or an enzyme like HRP or alkaline phosphatase. You need to use two different, and you want to label a protein, you need to have two different reactive groups so that you aren't cross-linking your protein. And so in this case, we use, in this slide, in red, we're signifying that the antibody is the red circle with a deprotonated amine, and you have your heterobifunctional SMCC, and it also has a reactive maleimide on the end of it. So you just do the reaction with the ester. It forms an antibody with a reactive maleimide. At the same time that you're doing that reaction, you can also have your thiol created by cleaving a disulfide bond in your protein, and then you reduce it with DTT or beta-mercaptoethanol or TSEP, and you purify that, and then you mix the two together, and you have the reduced thiol is able to react with the maleimide group, and that forms a stable thioether. And that's a really good way to make a protein-protein conjugate. It's a little bit more technically sophisticated than people that use the next thing that we're going to talk about, which is EDAC, or carbodiimides. So people use this a lot. It's very common. So if people have two different groups on proteins that they want to modify and make a conjugate, they'll use something like EDAC, that's a crosslinker that you can convert a carboxylate to a reactive ester, really, and then react that with your amine, forming a stable thio bond. And the thing that is interesting is that when people are using EDAC, a lot of times, because it will form this N-acylurea after it goes through the reactive O-linked acylurea. And the problem with that is that that's non-reactive. So it says it's stable because it is. It won't react. So a lot of times when people are using EDAC, they'll also add something like an NHS to cause it to be a little bit better, more reactive way to get to a cross-linking. This is common. It's kind of a sledgehammer approach, because when you're doing a EDAC or something like that, there's always that possibility that you're going to cross-link the protein to itself. Because most proteins that have a carboxylic acid group also has amines. So if that's a possibility, it's not necessarily the most sophisticated way to go. But it definitely works, and people use that for making things like our carboxy microspheres or quantum dots. That's a good way to make a conjugate where you don't have any reaction with amines on the microspheres or quantum dots. And one of the last ways that we're going to talk about is periodate oxidation. And this is really common where people have a carbohydrate or a reactive group that they want to modify or create a reactive group, and they use periodate oxidation. It's pretty easy to do, but it's not super robust. So a lot of people that have tried it, you know, it's difficult to get the same reaction over and over to get a common conjugate when you're trying to do this, but it works. It's a good. And I did notice that we do have a protocol online that you can see that tells about how you could do it if you needed to know, if you wanted to do this. And the basic way that you do it is you're oxidizing your bisphenol diol, and you're creating an aldehyde or a ketone, and those can react with your amine-containing dye. And then the last methods that we're going to talk about for labeling your protein is click chemistry. So in many cases, people want to just be able to label their protein, or, you know, if they're going to do a Western blot or something, and they want to be able to see, you know, that they have the protein that they're looking for. You can do this click chemistry where you're using a copper-1 as a catalyst for forming this alkyne azide reaction, and it forms a stable triazole conjugate. The thing that we don't like about this is that if you're doing it with a protein, some antibodies, some proteins like RPE, they do not like to be in the presence of copper-1. It's rather cytotoxic. So there's another type of click chemistry called the dibo click chemistry. This is a modified reaction. It's still an alkyne, but it's a dibo, so it's a stressed linkage. So it reacts the azide and the alkyne react and they form the stable triazole conjugate, but it's done without the use of copper 1. And so it takes longer, but you also don't have the problems with the cytotoxicity that you would have if you used copper 1. So those are both quick chemistry, and they both work. They're bioorthogonal, so you don't have any side reactions. They're very specific, and they're very stable. So moving on, so we've basically covered the overview of the different ways that we would do the labeling. Now we're going to talk about the different types of kits. So as we talked about before, you have to ask yourself those questions. So how much do I want to label? Is my protein purified? Is it an antibody? Is it some other type of protein? Is it in PBS? Does it have stabilizers? Those are all really important things to know. And one thing that we like to make sure that people realize is that one of the things that a lot of people tell us when they have problems is that they say, you know, oh, I got it from somebody in a lab down the hall, and it's in PBS, I'm sure of it. And a lot of times it's not. It has some other primary amine-containing part of its buffer, and that can really throw off your labeling reaction. So you want to be sure that you don't have any amine-containing buffers in your reaction, because that'll definitely throw off your reaction. So, and then again, how much do you want to label, and if you're going to use it to optimize or just, you know, what purpose do you have for doing your labeling? So looking at the tools, so if you have a protein and you have a small amount of protein and you have a contaminating protein like it's in a ascites or it's got BSA as a stabilizer or gelatin, you wouldn't be able to use some of our kits, but we do have ones that you can use to do labeling where that's not going to interfere. So if that's the case, and a lot of antibodies do, especially ones that come as really small aliquots, people have a problem, and you don't want to have to do a purification prior to doing your labeling, especially when you have a really small amount of your precious antibody. And so it's really a good idea to really think about what you need to do with it. So we have a couple of different options for doing labeling when you have competing amines. Our first option is the Xenon Antibody Labeling Kits. And these are really nice because it's a really super quick, super easy. You basically have an FC that you're able to use that's already labeled, an anti-FC, So it's an FAB fragment that you mix together with your primary antibody, and then you are able to use it immediately. And the next slide shows a diagram of really how that reaction goes with the Xenon labeling kits. So you have your primary antibody that you want to label. You just use the FAB fragments that are specific to the FC portion of your IgG, so that they're not going to bind in the binding site of the FAB part of your antibody. And then you use a blocker, and you're basically ready to go. You have a labeled antibody. It's not always the right option because it does have an off rate. So that's something that people need to be aware of. If you're going to do something where you're looking at a short incubation and you just want your answer, this is a really good choice. It's really easy. It's really efficient. It works very well. But it's not a covalent bond. It's a affinity, and that's why you have an off rate. If you want a covalent bond, we have our Apex Antibody Labeling Kits. These are designed specifically for antibodies. They do from 10 to 20 micrograms. You can do your labeling in the presence of BSA or tris or glycine or any of those kinds of things that are really common in small amounts of antibodies. And these are a really handy way to do your labeling and be able to get a product that you can use right away. So if you're going to do if you really want to check and see, like, how is this going to work? Am I going to be able to use this in my application? This kit really works very well. And we've heard a lot of people say that they were really happy to be able to have that option because you can, if you're trying to purify a really small amount of antibody and then do labeling, you can have horrible losses, and this is a really fast, efficient way to go in comparison to doing a separate purification and then labeling. So those are two options that you can use if your protein's not purified, if you have other interfering amines. And then for purified proteins, we have different products that you can use. So for purified antibodies, we have several different kits. And again, this is not specific just for antibodies. So the interesting thing about our kits is that we have two that are designed where if you use the antibody, we have a protocol that it tells you, what concentration to have your antibody in. You basically have your antibody, you add your bicarbonate to deprotonate your protein, you add it to a vial of dye, you let it react, you purify it, you're done. You don't have to do any kind of calculations. It's really nice. There's no, you don't have to do anything besides just mix and then purify. And again, like I said, it's not even necessary for a lot of these if you're doing something like flow cytometry or ICC where you're doing washes. You may not even care about getting rid of your free dye. But if you want to be able to do something where you're knowing what kind of labeling you're getting and you need to do optimization, we give you the option to be able to calculate your degree of substitution or your fluors per protein and know exactly what you've made. So the protein labeling kits are designed for 1 milligram of antibody, but our tech support will actually give you some advice if you decide that you want to try to use a different size protein and not necessarily an IgG. They're all composed of a vial of reactive dye. Actually, for this kit, there's 3 vials of reactive dye. Column chromatography resin, which is a 30,000 molecular weight, and buffer. So you basically have all the components and the bicarbonate. So you basically just can use the kit and you end up with your purified labeled protein. So this is a schematic that shows really just how easy it is. You have your protein, you add bicarbonate to your protein solution. You take that solution, you add it to the vial already measured for, if you're doing an antibody, you just add it to the reactive dye tube, you incubate, and then you purify. And like I said, that incubation step after that, if you want to go directly to use it, if you're doing something where you have wash steps, we've seen that it's really not even necessary in a lot of cases unless you need to know. And a lot of people want to know because they're worried if something doesn't work in their experiment, it's always easier to know what went wrong as you go along. And so we like to have that as an option, that if you really want to know, if something's gone wrong, if you didn't get the labeling that you thought you did or that you needed, or if it was over-labeled, it's always nice to be able to have that ability to check to make sure you're not going to waste your time downstream. And the next size kits we have, they're currently called monoclonal labeling kits, but these are really kits designed for 100 micrograms of IgG. Again, these have to be purified protein. They can't have any kind of other BSA or things because those will compete in the reaction. So BSA, for instance, has amines that will be deprotonated if you add bicarbonate. And so that's something that you really need to be sure that you know, that you don't have any kind of stabilizing proteins or any other kinds of amine-containing buffers in your antibody when you're using these kits. So basically, this has 5 vials of dye. It's designed to do 5 by 100 micrograms of IgG, and they come in many different kinds of Alexa Fluor dyes, many different colors. I will say that we are currently in the process of doing a name change. It's going to be the same kits, but they're going to be more specific so people will know that they're not specifically for monoclonal antibodies. You can use these for any kind of polyclonal, any kind of IgG works just fine. It's not specific that it has to be monoclonal, and I think that's been something that people have wondered about. Is monoclonal only really? I can't use a polyclonal? Yeah, you can. So it's just specific, and so I think that name change is going to be happening soon. Again, a really simple procedure. This diagram shows you have your protein at a set concentration, again, with no amines in your buffer. You add your bicarbonate to deprotonate. Then you take that solution, you add it to your pre-measured amount of dye that's already in the vial. You incubate, and this diagram says 1 hour, but just like the other diagram that we looked at said 15 minutes, you can also do this. We've looked at the reaction kinetics of the SEs or the esters that we use in our kits, and they're super fast reactions. Usually 90% happens before you even can do your measurements. So almost immediately the majority of your reaction has happened. So at 1 hour, you're going to have, maximal efficiency of the reaction, but you can do it in less time, and you'll still get the results that you need in most cases. And then in this case, we have a spin column where you do the purification. That's very fast. People like it, not a lot of hands-on time. But again, in certain cases, you may not even need to use that spin column if you just need to get your protein of interest, if you need your antibody labeled and you're going to use it in a downstream application where you just want to know that it's labeled and you want to assume that you have 100% of your antibody recovered, this is a great option to just go ahead and use it as it is. And then we have our next kit is our microscale protein labeling kits, and these are not designed specifically for antibodies. So with this kit, you get a kind of a guide that tells you what the amounts of protein, what the recommended amounts of dye that you want to use for the molecular weight or how big your protein is, and then we give you advice on how much dye you should use to try to get an optimal labeling. So anything that's larger than 12 kD is perfect for these kits. You can go higher than an antibody also. I think that basically what this kit does is it makes you do a little bit of math because you have to calculate what the amount of dye that you want to use to try to get, but we do give you guidance on that. Again, just like our other amine labeling kits, this also must be free of competing amines, so no BSA or gelatin, and no glycine or glutathione in your buffer system. These also are available with Alexa Fluor dyes and biotin. And this is pretty much the process that you do where you're doing the purification. These again use these little spin filters. So in these kits, we provide you with the resin, the dye, the purification. I guess these are actually not even called columns. They're more spin columns, but spin filter columns. But anyway, you get, again, a purified product at the end that you can measure your degree of labeling and know exactly what kind of reactivity you got. So you can do it to use it to optimize your procedures. So basically, when we look at our protein and monoclonal antibody labeling kits, these are all, those two specifically are set to make it very easy if you're doing antibody labeling. You raise your pH, you take your antibody bicarbonate mixture, you add it to the vial of dye, you mix it, let it react for a little while and purify it, and you're going to get something that's going to work in your application for the most part. And then again, like I said, you can optimize with these kits. So for the monoclonal antibody labeling kit, you use 1 milligram of antibody per reaction. You can do up to 3 reactions, so you can basically see, if I had over-labeled in my first reaction, I might use more protein when I do my second reaction to try to get a little bit lower degree of labeling. Also with the monoclonal kits, it gives you 5 different reactions, so you have 5 separate vials of dye. So if you have something where you're really trying to optimize, you can do your first reaction and then make adjustments based on what you want. And then I've talked about this quite a bit, but again, this is something that we know from talking to people that, absolutely are against using columns because they don't want to risk putting their precious antibody on a column and they possibly losing their protein. You can also, for a lot of cases, use no column, and you get really a convenient way to make a conjugate with great results. What we've seen is that compared to a lot of our competitors' kits, even without that don't use a column, our kits give results that are very, very good in comparison. But we also give you the option that if you need to know what you're doing, if you need to know, if you can't tolerate having free dye in your assay, you also have that option. Another way that people make conjugates with our kits is using the quantum dot labeling kits. So the Q dot nanocrystals are a pretty big seller. They're something that people really like because they have multiple colors off the same excitation source. So these are all UV excitable, and they have emissions ranging from 500 to 800 and a little bit beyond. We have a kit that you can buy. It's very similar to the protein-protein cross-linking that we talked about before, where you have the SMCC on the quantum dot, and then you reduce your antibody with DTT. You do your purification, you mix them together, and you have a stable conjugate. So there's been people that have had problems with using these kits. Some of the tips that people want to know are that if you start with your antibody at a higher concentration than the protocol tells you initially, you can actually get a more efficient conjugation, and that makes a lot of sense to try that if you're having any kind of difficulties. If you're using DTT to reduce your antibody, you need to know if that's something that your antibody can tolerate. Sometimes it can't. If that's the case, you might need to find a different way to go about creating the free thiol. We do recommend an option for that is using Trout's reagent or 2-aminothiolane. That will give you basically going from an amine to a free thiol directly. In that workflow diagram, and I'll go back to that slide, you would just substitute from the DTT for your antibody. Instead of that excess DTT, you would just use 2IT and then do the purification, and it's still going to react with your SMCC-modified quantum dot. And then another thing, when you are purifying your conjugate that you've made with the quantum dot labeling kit, is that you definitely need to follow the directions. So you have to, there's a column at the end, and in this case, because it's basically doing a protein-protein and you don't want to have free antibody around in your reaction mix, you do want to use that column for your final cleanup. And you do want to start collecting the drops when the color appears, but you don't want to collect more than 10. A lot of people have problems with that because I still see my product in that column, and I don't want to lose it. But if you do that, you also run the risk of collecting some of your unlabeled antibody, and that can interfere with results. This is the emission spectra of our Alexa Fluor dyes. These are our signature dyes for molecular probes. They're very bright, they're photostable, and we have a wide range of dyes that can fit most people's needs. Depending upon what you're doing, we have many dyes that can be used. I would like to point out that on this list it has Alexa Fluor 680 and Alexa Fluor 790. Right now we don't offer an Alexa Fluor 790 labeling kit, but we will. In August, we're going to be launching a new 100 microgram size or monoclonal size kit with the 790 and also adding a 680 labeling kit. So for people that like to do Western blotting and things like that where they want a long wavelength dye, we're adding to our collection. So those will be coming soon. And so this is probably my favorite side of this whole presentation because it gives you a really quick guide to figure out what are you trying to do and can you use our kit with your protein. So it goes through and it basically talks about how much protein do you have and does it have contaminants. You know, it's a dichotomous key really, so yes, no. If it's purified, you can use it, and even if it is purified, you can still use our xenon labeling kit. It's not necessary that you have a protein that has any kind of stabilizers to use xenon. For a lot of applications, people like it for what they're doing, especially if they're doing something like flow where they need a quick, easy answer, and there's not a vendor for their labeled primary antibody. This is a really good way to get your labeled antibody and get a quick answer. And then, again, you've got the Apex, which gives you the covalent bound dye to your antibody, and the other ones also everything else below that. So the protein labeling kit and the fluoroporter, the monoclonal antibody labeling kit, the microscale protein labeling kit, and the Q-dot antibody labeling kits all give you a covalently bound label. So they're not going to come apart over time. So that's important if you're going to do something where you have long incubations and you can't handle having an off rate. You wouldn't want to use xenon in that case, but all of the other kits that are offered do give you that covalent bound dye. And then we also look at do-it-yourself. So we don't just sell kits. We try to make the kits to help as most people as we can to make it easy to do the labeling. But we also, for people that they know what they're doing or they want to do a larger amount and they want to do fully optimized, or they want to make something that we don't necessarily sell, we have dyes and haptens in amine reactive. We have maleimides. We have all sorts of different tools that you can use to make your own conjugates. We sell cross-linking and reducing agents, so you can SMCC-modify any protein that has an amine. You can add thiols to a protein with SPDP. It reacts with amines and creates a disulfide bond that then you can reduce and use free thiol. So these are tools that we give you so that if you need to make something like you have your specific antibody, your primary antibody, and you need to have it with alkaline phosphatase, we at least can give you some guidance with doing that and making that for yourself if one of our kits doesn't work for you. We also sell RPE, that's pyridyl disulfide derived. So you have basically a disulfide bond that you can easily reduce and use for making like a protein-protein conjugate with RPE. We also sell biotinylated reagents. So if you want to use streptavidin for doing a signal enhancement where you're looking at a amplification of your signal, we have things like that. We also sell different avidin, streptavidin, neutravidin conjugates. So if you're biotinylating something and then want to be able to see it with a fluorescent dye, we also have that. Again, we sell quick reagents. So we have azide, alkyne, and dibo-reactive dyes. We also have different groups that you can buy. If you want to pegylate something, you can do it with some of these things. And then we also have the Q-dot ITK kits, where you can have a carboxyl or amino peg quantum dots. So you can actually make whatever you want. You can do, the Carbodiimide using EDAC and make your own conjugate with that. And we also, anything that you can think of that you want to make, for the most part, we usually have things. We don't always have it in kit format, so it's always nice to know that we're here for you. If you want to make a conjugate and we don't have a kit for you, we can try to help you figure out how to do it and what the best way to go about. Let's see. So, tips and tricks. So, this is my number one, and you're probably tired of hearing me say this, but if you are not sure what your protein is in, you always want to dialyze it versus something like PBS or some type of protein buffer that you know doesn't contain any free amines. So it's really common that people tell us, no, I'm sure I know what it's in. I got it from the guy down the hall, and he told me it was in PBS, and then it doesn't work. And so we've had a lot of people send us their protein. They say they couldn't label it, and the first thing we always do is we actually dialyze it, and 90% of the time that solves that problem. So if you're having a problem with not being able to label your protein, you can do a quick spin through a column that's equilibrated with something like PBS, and you've got your protein in the right buffer. Another problem that people have sometimes is having their column loaded. So they're waiting for that first strip of their column to start packing, and it never comes. And so our recommendation is that you can actually apply a little pressure to get that flow started. Some people like to use a little bit of water before they even load in their resin, or you can just simply, put your finger on top of the column and press down, or use a bulb and apply a little pressure just to get that flow going. And once it starts, it's going to pack and everything will be fine. But that's a problem that a lot of people have had. And then again, some people have problems where they see free dye coming off their conjugate. And it's not that the dye isn't covalently bound. Sometimes there's a little bit of free dye that kind of associates with the protein, especially if you have a hydrophobic dye, where it'll kind of stay with your protein, but it's not really bound. A good way to avoid this is to have your conjugate sit for 48 hours in the refrigerator, and then you can re-purify it, or you can do dialysis. And those are pretty simple ways to get rid of free dye if that's been a problem. Other tips and tricks, and one of the reasons why I put this on here is it's startling that, and we have people at our own company where I'll order an antibody and I'll check it in the UV vis, and the concentration I get is totally not what the vial says it is. And I ask, I'll finally get to talk to the scientist, and I'll say, how did you determine the concentration of this antibody? And, oh, well, I did a Lowry, or I did a Bradford. If you have a purified protein, the easiest way is to just measure the absorbance at 280. For an antibody solution, a 1 mg per mil solution in a 1 centimeter path length cuvette would be 1.4. So that would be the extinction coefficient, and you can directly calculate the concentration. It's way easier than having to do an assay, and it's also pretty straightforward. And then again, you can also use the nanodrop if you want to just apply your protein solution and measure it. If you do use a nanodrop, just remember, if you're not using a program that does the math for you, it's a 1 millimeter path length, and so your absorbance would be 0.14 instead of 1.4. But that's a really good way to go about checking the concentration of your antibody before you do your labeling, because it will make a difference. If you don't check it, if you're relying on, but they said it was this, because some vendors of antibodies will, like I said, they will do a Lowry or a Bradford, and it won't be exactly what you would calculate it to be. And then there are other times that people get it from somebody else, and there will be contaminating proteins, and it's really good when you're expecting that your protein's at 1 mg per mil, and you measure that it's at 6 mg per mil, and you're like, wait, what's going on? So that way you can tell sometimes if you do have a stabilizer. So it's always good to check it, but for some vendors you can take their word for it, but I always like to check it first just to be sure that my concentration is what it says it is. And then again, and we touched on this briefly, that if you don't want to risk reduction of your antibody with DTT, Trout's reagent is a really good answer for that. So you're not possibly causing your antibody to be fragmented. You just directly convert amines into sulfhydryl groups. One thing to be aware of when you're using amine chemistry and you're using an antibody is those amines can be anywhere. So you are, at the mercy of are my lysines in my binding site? Are there only a couple? So we do see sometimes that can be a problem. If it is a problem and you label in your binding site, again, consider using xenon because you're only going to be binding at your Fc portion of your antibody, so you're moving away from the binding site of your antibody. And you also can use Apex because the way that that works, so it purifies your antibody by binding to the resin in the tip, and then you're labeling while your protein is bound to the resin on the tip, so you're actually making sure that you're not going to be labeling in the binding site of your antibody. So those are two really, really easy ways to avoid labeling in your binding site. And then we like to recommend that there's a really great book by Greg T. Hermanson, Bioconjugate Techniques. We used to sell it. I don't think we do anymore, but it's still available. You can buy it on Amazon. It's a really handy guide for any kind of reactions that you want to do. And then finally, if you are just at your wit's end and you think, you know, I just don't want to take the risk. I don't want to do it myself, or I've tried it, it doesn't work. We also do custom conjugates. So that's a service that we offer where you send us your antibody or your protein, you tell us what you want labeled with, and we'll do it for you and send it back. Sometimes it's a simple fact that, you know, we'll discontinue something that, you know, 20 people maybe buy, and we can't support selling it as a catalog product. We'll sell it to you as a custom. We can make it for you. Sometimes it actually works out to where you can get more material by doing it as a custom. And then if you're not sure what label you want to use, if you have a certain excitation filter and emission filter that you're going to use in your assay, we can help you decide what dye is best for you, and we can give you confidential advice. We don't need to know, you know, all the specifics of what you're doing. You know, more information is better, but, you know, we can give you advice on what's going to work best for you. And then if you have grams of protein to label, well, obviously you're not going to want to try to use a kit, you know, that labels 1 milligram if you're going to try to be labeling 50 milligrams. It's just not feasible. It's not cost effective. So you can send us your protein and have us do it for you, or you can use our tech support to get some help if you want to try to do it yourself with one of our standalone dyes or biotin or something like that. We're more than happy to help you. So really, final slide. So, and I know this sounds pretty corny that we succeed when you succeed, but I think we really feel like, you know, if we can help you, you know, and we can get you to do your experiments properly, and if you have a good experience, it's a good experience for us, and we want to try to help. So, I think Olivia has answered any questions that you've had. I thank you for your time. If there's anything that you wish to know or have any questions, this will be repeated again this afternoon. This afternoon at 4 p.m. Pacific Standard Time, or no, not Pacific Standard Time, Pacific Daylight Time. And that will be at 4 o'clock. So hopefully you've gotten something out of this, and thank you very much.